Good morning. This is Mr. Handy Clement. Thank you for watching us. Thank you for watching this video again and this live again. This is Year 9, Week 2. Today's topic is about photosynthesis. Photosynthesis is a way that the plant makes food. The plant is a producer. It's the starting point of the food chain. Just like what we learned yesterday, I introduced a concept which is carbon sink. It's a process that the carbon from the atmosphere, becomes foods, become the starch in the plants. Okay, so today we will go to the tail of the leaf to see what is the structure of the leaf. So this is a leaf, and this is the upper layer, and then this is the bottom. The upper layer, usually when you touch on the surface of the leaf, there is a waxy layer. The waxy cuticle, this waxy cuticle always try to reduce water loss, because this is the place where facing out and direct sunlight. Our waxy layer will help to reduce water loss, and this I specially made for pink color. This is so-called one cell thick upper epidermis. This layer of the cell looks horizontal and look thinner this way, as the light can shine through this layer. For example, this is light. The sunlight can shine through this layer and reach the second layer of the cell. This is the idea. So this layer is transparent. However, this layer also have one function, which is to prevent the entry of the disease-causing organism. So this is a protection layer, which is called upper epidermis. Epidermis means outer layer of the cell. And then here we come to the, look like bricks one. This is column arrangement, very narrow arrangement, very tight, close to each other arrangement. This is called parasite mesophyll. Parasite cell. This is to maximize the absorption of light. Can you see? All this green color, are chloroplast with chlorophyll inside. So all these this is a nucleus, and this is a small vacuole, and all these chloroplast, all these chloroplast, will try to absorb as many as possible of the sunlight energy, to produce foods for us, autotroph. And this part, I try to make it around. This is so-called the vein. From the leaf, a leaf, you can see the vein, right? Let me draw here. This is a leaf from outside. So this is the vein. So now, this picture means that I cut here. And then you can see there's a vein, there's outer layer, everything. So this is the idea. The xylem is for the water transportation. The water from the roots will go through the xylem, root hair xylem, and reach to the top, which is the leaf. And phloem is the organic compound glucose transportation. Can you see I try to make a very light blue color to indicate this is the water. Okay, as compared to the very tight arrangement there, this is loosely arrangement. Okay, and a lot of water and some air will through here. Okay, some air is here. Okay, so this is a thin layer of water, and water diffuse out. Okay, water diffuse out just like yesterday's year 8 week 2, or through a process called transpiration. Okay, there's a door here. The door cell is called stomata. Okay, this control the in and out of the water and also exchange of the gas such as CO2 and O2. Okay, so from these two, okay, if this we have another gas cell here, stomata. Okay, as a product of the photosynthesis, there will be a lot of CO2 here. O2, sorry, O2. Okay, and the outside will have a lot of CO2 as compared to the inside environment. So these two will exchange, okay, diffuse out. The high concentration of the oxygen will diffuse out, whereas the high concentration of the carbon dioxide will diffuse in. And the carbon dioxide go in, combined with the water, will produce carbon dioxide plus water. These are the material we need. Will produce C6H12O6 plus O2. Okay, O2. Okay, so this is carbon dioxide plus water will produce glucose in the simple form and oxygen. This one is not. Okay, this one is not. Okay, wait. Okay, this one is for this one. Okay. Okay, enlarge the cell for you to have a detailed look. Okay. This is Mr. Handy Clement. Okay, cell surface membrane. Okay, wait, let me see. Okay, the outer layer is called this one. This green color. Okay, this is the wall, cell wall. Okay, and then when you take out the wall, there's still a membrane cover this yellow color. This membrane, this whole thing is called cell surface membrane. And we have this look like a lake or pool, right? This is Foucault, which is water to help for the water. If this is full of water, they will give you a support, turgid. Okay, if this is no water, then the plant will look like flaccid. Okay, so all this green color, okay, is chloroplast containing chlorophyll. Okay, this is contain most in parasite. Okay, parasite have the most of this chloroplast. If the mesophyll, there's contain only 1 or 2. Can you see? Less. Okay, there's almost none for the upper epidermis. Can you see? This is none because it will block the sunlight. But here there's a lot, and the arrangement is become thicker and deeper, so that can trap the maximum light possible maximum light. Okay, so plants make glucose and store them as starch. Okay, this is long chain. This is only one. This is only one. This is, for example, this may be C6H12O2 or O6. Sorry. Okay, but this one is not. This one may be C6000 H12000 or 6000, for example. Okay, so this is such a long chain. Okay, they combine together. This is only one, for example. This is for glucose, and then for starch will be like very long. They combine together. Endless. Can you see? Endless. The next paper will be plant deficiency. Okay, this one especially for you. I copied from somewhere, and make it suitable for my teaching. Okay, this is plant deficiency. Okay, so this is the upper new leaf. If the new leaf misshapen, can you see stunned? Okay, that means lack of calcium. This one no calcium. Okay, and with no iron, that means the young leaf will look yellow and white, not green enough. Okay, and potassium. Okay, if you lack of potassium, okay, the outer side and the edge will be yellowish. Okay, inside still okay. This is no potassium. Nitrogen. If no nitrogen, you see, is very green light for the upper layer and the yellow at the bottom layer. Okay, and manganese. Okay, if you don't have manganese, there's a yellow spot and elongate holes. Okay, see there's a hole here. Oh, between veins. If without CO2, yeah, that means this is this starch plant already. Okay, white deposit and stun. As you can see, no photosynthesis happen here. Phosphate. If no phosphate, the leaf look very darker than normal, and you'll experience loss of leaf. Magnesium. Yellow from the outside, in, and vein remain green. This is the deficiency of the plant. Okay, all this material this simple material I upload it on Google Drive and also in Facebook, called Handy Tea, I think so. Handy Tea, Handy Tea. Okay, this is for your learning purpose. It's not for reselling. Okay, and for the fertilizer, we have NPK. Okay, artificial fertilizer. Okay, N means for nitrogen, which is always related to the protein. Okay, and phosphorus. Okay, as you can see here. Okay, help to have prevent loss of leaf. Okay, and the potassium is quite important here as well. Okay, to help to the plant to become healthy. Okay, this is the plant efficiency. For the last one today, okay, I only introduced some of the important parts for this syllabus, and one still need as my introduction. I have done it in the first week. Okay, you still need one still need the textbook to be the main thing. Okay, for your study. Okay, number one, you still need to do textbook. And then you may listen to my framework and selected topic just like today's. And number three, you may make your own notes just like me. Number four, you must do your division, articles, other source any other source that you like, YouTube. Number five, some past paper. Okay, if you face any problem from all this, okay, you can always contact me. I will try my best to help. Thank you very much. Okay, so today I have explained this one in my drawing yesterday, but today I will try to use this book. Okay, to demonstrate the thing, you need to have this start plant. This part means that you need to put the plant in the cardboard or box for 48 hours. Okay, some book says different hours, but the important is I think 2 days. 2 days. Okay, then the plant will use up all the starch available in the body. Okay, so this is the way we we do the iodine test. Okay, the very first, the keyword is boiling water to break the cell wall. Okay, for 2 minutes, so the leaf will become very soft, and then the enzyme also stop the enzyme of the activity of the enzyme. Okay, and then next, we have warm water bath. Okay, so you have warm water, and then inside the test tube you put the ethanol. So we are using ethanol, okay, to break the chloroplast, okay, and not the water. Water is just outside. Okay, so the chlorophyll will come out, so the leaf will look pale. Okay, and then you need to rinse with some warm water to remove the excessive ethanol, and then place on the white type. Okay, white is important because white type can let you to see the color, not other color, please. Okay, then the iodine solution is added. Okay, originally without starch, the color should be orange brown. If there is a starch in the leaf, the color will look blue black. Okay, which means that the positive result is blue black if there is the presence of the starch. Okay, and then to test about there are many things here. Okay, for A, that means cover with black paper. Okay, both sides. That means or aluminium foil or anything that is opaque. That no light can access to the part. Okay, and you test. This is a discharge plant. After I think let's say for 1 day will do. Okay, after 1 day you take the leaf and test using the above method and test, and then you can see the part with the sunlight. Okay, there is blue black color, which means that the plant did mix some starch and the bottom part as well, but not in the middle part because this is covered and the chlorophyll here cannot make any starch. Okay, the next B. The B you have two. Oh, this is silver. Okay, this is solar lime. This is hydrogen carbonate solution. Okay, this is to absorb the CO2, means that the environment is without the CO2, and this is the environment with the CO2 because this release a lot of CO2. Enough one. Okay, so without CO2 you look white and pale, and then after test is still orange and brown without changing any color. But this one, we have a lot of CO2, the whole leaf will turn blue black, which is positive result for the iodine test. Okay, the last one. If this is the leaf, you can see at the edge, this is the one type of leaf that there's some color. This is not the whole leaf is green color. There's a white color at the edge. Okay, so variegated leaf. So only the green part, which the part contain the chlorophyll, will produce the starch. So when you test the whole leaf, the outer layer, which is the white color without the chlorophyll, will not have the starch. Okay, this is a very brief lecture. I hope that you like it. Okay, you can take this as a preview. Always like us and share us to motivate us, because online teaching, we need your motivation to share and help us to get as many as possible, so that we can know that what we provide is that you really like it. Thank you very much for our friend that already like us and share us. Thank you very much. Okay, and this is for today. I hope that you enjoy this one, and see you later at 10 a.m. for the mathematics year 9 math. Thank you very much. Thank you.